effects in different cell types. And as we're starting to see, this is probably due to differences in the receptor and differences in the pathway to which the receptor is attached within the cell. We talked about the various kinds of signals that cells are getting, either sort of all the time, things like survival signals, or every so often, like division signals, things like that. Because otherwise, if they stop getting, especially the survival signals, a pre-programmed process called apoptosis, essentially cell suicide, kicks in. And so we're looking at sort of the general properties here of a signal molecule binding to a receptor and being linked to some kind of intracellular process, usually a series of proteins that will have some effect on the cell. Changing metabolism, we'll look at that example today. Changing gene expression, changing cytoskeleton as a prelude to allowing the cell to move or something like that. Okay, so we had started looking at the different types of pathways in terms of the different kinds of receptors. So we had looked at an ion channel link receptor, that was the nerve cell example, and we looked just generally at the enzyme link receptors, and we'll spend much of today looking at this third type, the G protein link receptors. So we looked at the example for an ion channel link receptor of nerve impulse moving from one nerve cell to a second nerve cell across a small distance between them called the synaptic cleft. And so that was an ion channel linked situation in which, as the action potential, this wave of sodium ions reaches the terminal knob of one nerve cell, that triggers a calcium channel to open. Calcium spills in, that causes the neurotransmitters to be secreted, and there's our signaling molecule. They're going to bind to ion channel link receptors on the next nerve cell, open them up, sodium rushes in, and there's your action potential in the next cell. There we go. Okay, so here's an example with acetylcholine specifically binding to this channel. It opens it up, sodium comes in, and we get a new action potential going there. So I would like you to know this example in the detail that we looked at it. The catalytic receptors, we weren't going into too much detail here. What I need you to take away from this is that they have an enzymatic activity, typically a kinase activity, a phosphorylation activity, and they typically have to dimerize in order to work. So looked here at the example of the epidermal growth factor, the EGF receptor, and so when it gets bound by the signal molecule, two of them have to get together, they phosphorylate each other, and typically on tyrosines or serines or threonines, and then these phosphorylated sites are typically sites of sort of attachment for other relay proteins or signaling proteins that continue the process. We're not going to, I don't need you to memorize a specific pathway. We looked at part of the RAS pathway here is just a quick example, but you don't need to memorize this particular one. And then we're just about to start on the G protein link receptor, the kind of animation for the catalytic receptors that I want to just show you.
Growth factors signal cells to grow and divide by acting through tyrosine kinase receptors. Binding of signal molecules causes two polypeptides to join, activating parts of each that act as tyrosine kinase enzymes, which then phosphorylate tyrosines in the tail of the other polypeptide. The receptor protein is now recognized by specific relay proteins inside the cell. One tyrosine kinase receptor may activate several relay proteins at once, triggering several different effects with growth factors signal cells to grow and divide by acting through tyrosine kinase receptors. Binding of signal molecules causes two polypeptides to join, activating parts of each that act as tyrosine kinase enzymes, which then phosphorylate tyrosines in the tail of the other polypeptide. The receptor protein is now recognized by specific relay proteins inside the cell. One tyrosine kinase receptor may activate several relay proteins at once, triggering several different effects within the cell.
So again, that's pretty much what I need you to take away from that. But again, we're not going to look at a specific pathway for that type of receptor. Okay, we'll pick up with these in just a second. How are we feeling about ion channel linked receptors and catalytic receptors, generally speaking? Lovely. Okay, our third and final type of receptor is called a G protein linked receptor, because they are going to work through proteins called G proteins that we'll look at in a second. The receptor is shown here. Often the G protein linked receptors are what are called 7 transmembrane receptors, because they cross the membrane 7 times. 1, 2, 3, 4, 5, 6, 7. And so they would need to have at least 7 of those stop transfer sequences that we've studied before to get all of those sections inserted into the membrane way back in the ER, and eventually they'll make their way to the cell membrane. And so they often have this 7 transmembrane configuration, and again they have usually a substantial cytoplasmic section here that will interact with the next sort of piece. And that next piece is often a G protein. And they're called G proteins because they interact with the nucleotide GTP. So that should be hopefully easy to remember. Okay, so G for GTP. There are a couple of different kinds of G proteins. We're going to focus on what's called the trimeric version, because trimeric for 3 pieces. So there's an alpha, a beta, and a gamma piece to them. There's also a type of G protein called a monomeric G protein, and actually the RAS that we had looked at earlier. RAS is a G protein as well, but it's a monomeric kind. But for the couple of examples that we're going to look at here, we're going to look at the trimeric type. So at the sort of start, we'll kind of look at this in general, and then we'll look at a couple specific examples. Okay, so here's our receptor. Here's our G protein that's in the inactive state. Okay, so everything's off. Okay? And it's inactive because it has GDP attached, the diphosphate, the D, GDP form. So that's the off version. Okay. In comes our hormone, here in comes our signaling molecule, binds to the receptor. That's going to cause a slight shape change in the receptor, such that it can now interact with that G protein. And when the receptor interacts with the G protein, it basically causes something called nucleotide exchange. The GDP is going to come off. GTP, the triphosphate form, is going to come on. That's the on switch. Okay, so when the G protein has GTP attached, that activates the G protein. So this nucleotide exchange is sort of the on switch being flipped. And what often happens is what they're showing us here in the third part, and notice that it's the alpha subunit that has the nucleotide attached. So when GTP is attached to the alpha subunit, what often happens is that the alpha part detaches from the rest of it, from the gamma delta part, and one or sometimes both of those pieces trigger next steps. I think in both the examples I have for us to look at, it'll just be the alpha part that does it, but in some other examples it can be one or both of them triggering next steps. So that's kind of the on part, which we again usually pay most attention to. There is an off switch to this as well, which is the hydrolysis of that GTP. So eventually that GTP gets hydrolyzed back to GDP. GDP means it's inactive, it's off. Okay? So there is a sort of a time aspect built into this. Eventually that GTP gets hydrolyzed back to GDP. That causes the pieces to come back together, and now it's off again. Okay, so there is an off switch built into this part of the process. Okay, so again, this is why these are called G protein link receptors, because they're going to work through these G proteins. So that's kind of the general idea of how the process gets started. And then specific pathways then will involve specific G proteins with specific alpha subunits that trigger specific next steps. So we're going to look at two specific examples of pathways that use G protein linked receptors. But before we look at the specific examples, this general scheme kind of makes sense with the GTP and the GDP aspect. Okay. So this is sort of exactly the same thing. It's just a different. I took the picture from a different book, so if you like this version better, that's fine. But it's basically showing you the same thing as the previous slide. Okay. So the first of two specific examples that we're going to look at is called the fight or flight response. It's mediated through a G protein linked pathway and will end with a change in the metabolism of your cells. Okay, so your fight or flight response is sort of self-explanatory, but it's your response that you encounter something sort of scary or dangerous or whatever, and you have two choices. You can fight the thing, or you can run away from the thing. Both of them are going to require an increase in metabolic activity. Okay, so that's why they're sort of linked together. So this ends up being mediated through a pathway just like we've started to look at. So when you come across something scary, so you're out camping in the woods and a bear comes along, this is always my silly example, you're going to either fight the bear, hopefully not, or run away from the bear. That would be my preference. Okay, but it's going to be mediated through this pathway. Okay. So when you see the bear, a signal molecule called epinephrine is secreted into your bloodstream. Okay, so this is therefore what kind of pathway? Dump a signal molecule into your circulatory system. Regulated. Oh, well, I guess it's regulated secretion in that part, but what kind of signaling pathway? Right, it's going to be an endocrine pathway, right, because we're dumping the signal into your bloodstream, so it's going to have an effect elsewhere in your body, mostly on your liver and muscle cells. Okay, so you see the bear. Epinephrine is now surging through your circulatory system, and it is going to hook up with a receptor called the beta-adrenergic receptor. We're going to focus on your muscle cells. So pretty much like this first part. Okay, so let's pretend this is specifically the beta-adrenergic receptor. The green signal is epinephrine. Okay, so you have the epinephrine binding to the beta-adrenergic receptor. That again will slightly change the shape of the receptor so that it can interact with the G protein, and that interaction, as we've seen, go back to the pictures, causes nucleotide exchange. I'm just kind of summarizing that here. Okay. So the receptor interacts with G protein, causing nucleotide exchange. That nucleotide exchange is going to activate the G protein, right? In this example, it's the alpha subunit that will trigger the next step. And it is going to, as you see here, interact and activate an enzyme called adenylyl cyclase. And as you can see on the slide, that enzyme runs this reaction. It takes ATP, my old friend, and converts it into something called CAMP, or abbreviated CAMP, which is cyclic or cyclic, depending on how you say it, AMP. Let's show the next slide, I think. Yeah. Okay, so here's our old friend ATP. When adenylyl cyclase gets its hands on it, it chops off 2 of the 3 phosphates, then takes the third one, the third phosphate, and attaches it a second time to the sugar. So it creates a cyclic or a cyclic section here. And since there's only 1 phosphate left, that's the monophosphate AMP. So it's called cyclic AMP or CAMP. That cyclic AMP, or that CAMP, is known as a second messenger. What was the first? If there was a second messenger, there had to be a first messenger. What's the first messenger? Yeah, so our original signal is the epinephrine. We don't really usually call the first messenger, but in this context, we can. So that's the first one. Here's our second messenger that's inside the cell. And now it's going to be this CAMP that's going to trigger the rest of what happens. Are we good so far? Okay, so we have the epinephrine binding to the receptor binding to G protein, causes nucleotide exchange, activates the G protein, alpha subunit activates adenylyl cyclase, which runs this reaction and makes lots of cyclic AMP, our second messenger. Okay, what is that second messenger going to do? It is eventually going to result in glycogen breakdown, but that's going to take a few steps, as we'll see here. Okay, so this left part, again, is just kind of a summary of what we've done. So here's epinephrine binding to the receptor. Receptor activates the G protein. We get nucleotide exchange. The alpha subunit activates adenylyl cyclase. Adenylyl cyclase converts ATP into cyclic AMP. So here we are right here. Okay, so here's the rest of the pathway. So here's our cyclic AMP. It is going to activate or bind to and activate another protein called protein kinase. Okay. What do kinases do? They phosphorylate things. Okay. So we've started seeing that already. Here's going to be another example of that. So when cyclic AMP binds to protein kinase, it becomes active. It wants to phosphorylate something. What it wants to phosphorylate is another protein called phosphorylase kinase. We're going to, just so we can distinguish this a little bit better, what the slide is calling protein kinase is also known as protein kinase A, so that'll let us abbreviate it PKA instead, which will be better. Okay. Protein kinase A is going to phosphorylate, and that will in turn activate the next one in line, phosphorylase kinase. Just do that one It's another kinase, and kinases like to phosphorylate things. That active phosphorylase kinase is going to activate what the slide is calling phosphorylase B. That's also known as glycogen phosphorylase. That might be the better term for us to use. So if you want to make that change on this slide as well, instead of phosphorylase B, we're going to call it glycogen phosphorylase. And finally, what does glycogen phosphorylase do? It takes glycogen. Now let's remember glycogen. Oh my gosh, we have to remember all the way back to August. What is glycogen made up of? What is glycogen? Yes, it is sugar. That's good. Any more information about glycogen? What kind of sugar? What level of carbohydrate is it? It's a polysaccharide of glucose. See, it all comes back around. Right? It's a polymer of glucose. Right? Thousands and thousands of glucoses strung together. What glycogen phosphorylase is going to do is start clipping those off one at a time. So it releases, well, technically glucose 1-phosphate. We can just kind of think about it as glucose. So glycogen phosphorylase breaks down glycogen, glucose. Where do you think glucose is going to go? We want to either fight or run away from the bear. Well, we're already in muscles. So what's going to happen to glucose in the muscles? Pardon? Right. We're going to metabolize the glucose, right? Get the energy out of glucose and put it where? What does metabolism make for us? ATP. So now you have a bunch of ATP to either fight the bear or run away from the bear. The fight or flight response. A similar but not exactly the same process is happening in your liver as well, which is where you have also a bunch of glycogen stored if you recall. So, from that first messenger, epinephrine, the end of this pathway is a bunch of ATP to fight or run away from whatever it is that is troubling you. So it is a multi-step process. We have spent time. I've written all this out on the board. You've written it all in your notes. That probably means it's kind of important. Everyone see where I'm going with this, or is that too subtle? That's too subtle, isn't it? You need to memorize this. Okay? Between now and whenever our exam is. Tuesday the 17th. Okay. That was direct enough? That wasn't too subtle? Okay, good. Okay? I need you to know this step by step. Okay. Process kind of makes sense? The step-by-step process? Okay. Now, you're probably asking yourselves, why is this thing so long? Well, it's really not that long. If you study more cell-to-cell signaling, there are ones that are 3 times as long as this. So this one isn't, by comparison, isn't really that bad. But the more general question of why do signal transduction cascades tend to be long generally, the answer to that question is probably an idea known as signal amplification. To get from a tiny amount of signal molecule, little tiny bit of epinephrine, to a lot of ATP at the end, requires this idea called signal amplification. Several of these steps along this multi-step pathway get amplified. The signal gets louder. Okay? That if we just sort of jumped from, epinephrine binds the receptor and the receptor activates this enzyme, we just, you could build it that way presumably, but you wouldn't get the amplification between steps 1 and step whatever this one is that we do by building it this way. And that's what this slide is trying to show us. So not every step is amplified, but multiple steps are. Okay, so the binding of that signal, that epinephrine to the receptor, is obviously not an amplification step because my hand's the receptor and the marker is the signal. That's a one-to-one kind of thing, so that's not amplified. But it turns out that although we kind of only looked at it, as a one-to-one, that activated receptor can activate multiple G proteins. Okay, so there's not just one G protein in your membrane, there are lots of them. So that activated receptor can activate multiple copies of that G protein, which in turn can then activate multiple copies of adenylyl cyclase. So both of those are amplification steps. Well, that's not true. The receptor G protein step is an amplification step. The G protein adenylyl cyclase is not. That's a one-to-one. But we've already studied enzymes. Enzymes make lots of product, right? So it's not like each enzyme is only making you one cyclic AMP, right? They're probably making thousands. So that's definitely an amplification step. And then each of those cyclic AMPs can activate a protein kinase. Okay, so that's a one-to-one. But then each protein kinase can activate multiple copies of phosphorylase kinase, which in turn can activate multiple copies of the glycogen phosphorylase, which can then flip off hundreds or thousands of individual glucoses, which, if they all go through glycolysis Krebs electron transport, each one's making you 30-something ATPs. So, by a little tiny bit of signal, you can get thousands and thousands and thousands of ATPs by the end of this to, again, fight or run away from your attacker. So that's probably the reason that these pathways tend to be multi-step instead of just jumping right from here to here. There really wouldn't be an opportunity for this amplification. Okay, what we're seeing now, 3 or 4 of the steps at least are amplification steps. Okay, so that's probably the reason. So keep that in mind, that there is a good reason that these things tend to be so long as you're committing these couple of examples to memory for our final exam. Does that idea make sense, the idea of signal amplification? Okay, so that's one example. Here's your fight or flight response. Well, I really have that one in there, but this really isn't that important. This is, oh, I know why I have this in here. Because here's that protein kinase A again that we've seen as part of fight or flight. So in your muscle cells, that protein kinase A activates phosphorylase kinase, et cetera, et cetera. In other cell types, that same protein kinase A will activate other proteins, different proteins, which in this example here, so protein kinase A releases this protein here called C, which enters the nucleus, so it must have a nuclear localization signal, and that in turn activates a protein called Kreb, which is a transcription factor, which will change gene expression. So the idea here is that, as we've seen in other places, the cell sort of reuses the same components for different things, right? We saw, if you remember, during photosynthesis, we saw a lot of the intermediates from other metabolic processes in that C4 pathway. So here, protein kinase A can trigger multiple things, multiple pathways in multiple cells. Okay, so the cell does kind of reuse individual components in different ways. So that's really all we're talking about here. Okay. In the second example that I want you to know in detail, we're going to look at a different second messenger. Okay, so we saw cyclic AMP in this example. In our next example, we're going to see actually two more second messengers. One is going to be this sugar called IP3, which in turn will release a bunch of calcium, which will also be a second messenger. So we're going to look at an example that uses these two second messengers. We've done all this. Okay, the second example that I want to look at that is going to use these different second messengers is going to be what's called a slow block to polyspermy. So, when we're about to fertilize an egg, how many sperm do we normally want to do that? Just one. Right? But if you studied any kind of reproductive biology, you know that there's not just one sperm out there, right? There are usually millions, right? Or at least in the very, very high hundreds of thousands, right? All racing towards that one egg. Ideally, we only want one of them to win and actually fertilize that egg. Because if more than one do that, then we're going to have all kinds of crazy chromosome numbers, and that doesn't usually end well for the embryo. So there are some built-in processes that try to block that process. So to block many sperm from fertilizing. So we're going to look at the one called the slow block. If there's a slow block, there's probably a fast block, and there is. But that does not work through a G protein linked receptor, so we're not going to look at that one. We're going to look at the slow block. The fast block is nice because it's fast, but it doesn't last very long. So it happens almost immediately when the first sperm contacts the egg. It happens almost immediately, but it doesn't last for very long. The slow block, as we'll see in a second, takes a little bit longer to sort of engage, but it's sort of permanent. Okay, so this is sort of the permanent block to polyspermy. So it's going to work through a different G protein and a different G protein linked receptor and a different G protein, and use different second messengers. So that's why I wanted to show you this second example. Okay, so we're going to focus on this IP3 pathway as our example here. Here we go. I should rearrange these slides a little bit. Okay, so we'll backtrack. These are a little bit out of order. Okay, so here's our eggs. All of these sperm are racing towards trying to fertilize. Surrounding the egg is a layer of what they're calling here jelly. It's a layer of all kinds of things, proteins, lipids, carbohydrates, that sort of surrounds the egg. And the sperm sort of have to work their way through this jelly-like layer, sometimes called a vitelline layer. There's our egg color, and surrounding it is this layer I usually call it the vitelline layer. This slide's calling it the jelly layer. Same thing. So the sperm has to sort of work its way through this vitelline layer in order to get to the egg. And so it's kind of a race. It's a race to the egg, and it's a race to get through this vitelline layer to get to the egg, basically. So to do that, and we'll just go through this quickly, but it's a good reminder of cytoskeletal kinds of things, because what happens when the sperm comes into contact with that vitelline layer, it causes, well, a couple of things. It causes this little vesicle filled with enzymes to be released, to be secreted. And that in turn triggers a bunch of actin, a bunch of G-actin to polymerize into F-actin, which starts pushing the membrane of the sperm through this jelly layer. And so this section of the membrane that's being pushed out is called the acrosomal process. So we've just released all these all those enzymes, they sort of coat the acrosomal process, and those enzymes, as you can sort of see here, start breaking down the vitelline layer. So they sort of burrow a tunnel, essentially, through the vitelline layer as the acrosomal process is pushed through. And if it's the first sperm to get there, it's going to come into contact with the egg. The membranes fuse. Eventually, the nucleus, the male nucleus, sorry, purple here, will be injected into the egg for fertilization. So another part of this contact then between this, hopefully the first sperm and the egg, is also going to trigger this slow blocked polysperm. And this is what happens later. Okay, so if we go back a couple of slides here. Okay. This is sort of jumping ahead of the story, but the same initial kinds of events occur. So there is a signaling molecule on the sperm. That's going to contact the G protein link receptor on the egg. Now, this is a different modality than fight or flight. That was endocrine, with the signaling molecule is on the sperm and has to make contact with the egg. I kind of just gave you the answer, but what kind of modality was that? Right, that's contact dependent, right? The sperm has to make physical contact with the egg for this to happen. It is a G protein link receptor, so the same general things happen, right? That's going to activate the G protein is going to be activated through nucleotide exchange. And again, in this example, it's going to be the alpha subunit that's going to do the next step. This time, the alpha subunit, instead of activating adenylyl cyclase in the egg, it's going to activate this enzyme right here, abbreviated PLC for phospholipase C. It's a different enzyme. So obviously it's going to be a different reaction. PLC runs this reaction. It takes this particular membrane lipid, called PIP2, phosphatidyl. Make sure I get the name right. There it is. Phosphatidylinositol 4,5-bisphosphate, PIP2. Right? And basically it's going to cleave off, it's going to cut off the sugar part from the lipid part. The lipid part is diacylglycerol, DAG. For this particular example, that's not going to be important for us. But when it releases the sugar part, the sugar picks up another phosphate, so it becomes IP3, inositol triphosphate. This is what we're going to follow here. So let's just write that out here. So PIP2 goes to DAG plus IP3. We're going to be mostly interested in IP3, which is our first second messenger for this story. Okay, so far so good. So a signal molecule on the sperm coming into contact with the receptor on the egg. Triggers nucleotide exchange in the G protein. Alpha subunit is going to activate this enzyme that will run that reaction and create lots of this second messenger. So again, this is a amplification step to make a bunch of this IP3. All right, so what does the IP3 do? And if you like structures. Here are the structures of these things. I didn't memorize it or anything, but if you like having chemical structures and things, that's what it looks like. Here we go. Okay, so what is this IP3 going to do in the egg? Well, the IP3 is going to bind to an ion channel linked receptor inside the egg, specifically in the membrane of the ER. Remember I told you there was going to be another, we're going to come across another ion channel link receptor. Here it is. But this one's internal. This one's in the ER membrane. So when IP3 binds to it, opens it up, what comes out of that channel? A whole bunch of calcium that's been stored in the ER. So IP3 binds to ion channel receptor. In ER membrane. Releasing calcium. This is our other second messenger for this story. The first one was the IP3. IP3 opens up the channel. Out comes calcium. And what's the calcium do? Well, it actually does something very similar to what happens in the nerve cell. Remember, as that sodium was coming down, it opened up the calcium channels. What did the calcium do in the nerve cell? Yeah, it caused the release, the secretion of those little vesicles with the neurotransmitter. It's going to do something very similar here in the egg cell. It's not going to release neurotransmitters, but it is going to cause the secretion of the contents of these vesicles that are called cortical granules. So in the egg, a whole bunch of vesicles filled with all kinds of different proteins. Just going to draw a couple in. These are called cortical granules. And that calcium is going to cause secretion of the contents of these cortical granules. And that's what they're showing us here. So these cortical granules start exocytosing or secreting. So all of the contents then enter a little tiny space in between the egg and the vitelline layer. There's our vitelline layer, vitelline envelope. And what that ends up resulting in is having a whole lot of stuff right to the outside of the cell. So we're going to change the concentration of stuff. So now we have a whole lot of stuff here that we didn't have there just a second ago. What does water like to do when it encounters a large concentration of stuff? Yeah, it likes to flow towards that high concentration of stuff, right? And that's exactly what happens. So by really very dramatically, very quickly increasing the concentration of stuff right there, water rushes in. And what that water does is it essentially pushes the vitelline layer, the vitelline envelope, away from the egg. What's trapped inside that vitelline layer? All those other sperm that are trying to get to the egg. So if we're pushing it away, now any sperm that's still trapped in here can't reach the egg anymore. So it's kind of a clever way of doing it. And this is, like I said, a permanent solution to this problem, because once you push that away, there's no way that the sperm can get to the egg anymore. So it's kind of a clever way of doing it. All right, so let's finish up. So calcium causes secretion of cortical granules. Increase concentration. Which brings water. And the water pushes away the vitelline. With all the other sperm. So all the other sperm get washed away essentially, and ideally only one sperm got to the egg in the first place to trigger all of this. Does that make sense? Okay, so I like that example because it uses this other pathway, this IP3 pathway, and the calcium pathway rather than the adenylyl cyclase that we saw earlier. But they're both G protein linked processes. Obviously, two different signals, two different G protein linked receptors, two different G proteins, but the general features of both of these are similar. You should know this one in detail as well. Okay, so we looked at the slow block of polyspermy. There are other pathways that involve IP3 and diacylglycerol. So these are just some other examples of pathways we could have looked at. I like this one in particular. And this slide is just sort of showing us that very, very often in real live cells, different pathways are often interconnected. So if you study this, if you take my cancer class or my immunology class and we do some of these pathways in those classes, you see very, very quickly that what seem to be separate pathways are very, very often connected through common intermediates. Okay, so that's really all that this is trying to show us, is that very often what we've sort of been studying as separate things are often interconnected. So if you ever look at this in more, in more detail in future classes, you'll see that very, very clearly. And then very quickly, the last thing I wanted to mention are signals that are lipid-based. So we've been so far looking at signals that have either been proteins in the membrane or secreted molecules that were hydrophilic, okay, that couldn't cross the membrane, that had to have the receptor stuck in the membrane of the target cell. But we've seen that whole other category of signal molecules that are lipid-based, often cholesterol-based, the steroid hormones. These are lipids, right? So these should be able to cross the membrane of the target cell, and they do. So, very, very often, the receptors then for these steroid hormones are usually not stuck in the membrane because the signal can go across the membrane to the inside of the cell. So having the receptor stuck in the membrane wouldn't do any good in that situation. So what we see quite often, in fact most of the time, is that steroid hormone receptors are usually intracellular. Those receptors are floating around the cytoplasm. And that's what they're showing us here. Just in general, we're not going to look at a specific example, but just a generalized one. So here's a steroid hormone that can get right across the membrane because it's a lipid. It hooks up with its intracellular receptor, and these receptors are very, very often transcription factors. So when the steroid hormone binds to the receptor, that typically, again, changes the shape of the receptor ever so slightly. In this case, so that the nuclear localization signal is revealed, gets it across the pore into the nucleus, and very often these receptors are also transcription factors. They hop on specific genes and increase the expression of specific genes. So that's the other big theme of steroid hormone pathways, is that they very, very often result in a change in gene expression. So they turn on or off, depends on the specific example, specific genes. Because they have these intracellular receptors that work very often as transcription factors. So that's a big difference, fundamental difference between the examples that we've been looking at that have membrane-bound receptors and the receptors for steroid hormones. And this is sort of the same thing. This one's from your book. So again, cortisol, which is an anti-inflammatory. If you've ever had a cortisone shot, it works the same way. So it can cross the membrane, hooks up with this receptor, gets in the nucleus, triggers specific genes to turn on that have anti-inflammatory, in this example, anti-inflammatory activity. Kind of there. Okay. Okay, so that's a very little bit about cell signaling. Again, what we did in 2 days, you can take an entire course in, if you so choose, up in University Park. But just to give you a little flavor of it here. Okay, so we have 2 more topics left. We have cell cycle and mitosis, and we'll finish up the course with meiosis next week. In the meantime, we have lab tomorrow. We'll do our last lab on mitosis, so we'll be a little bit ahead of the game in lab. And you'll finally, after what seems like weeks and weeks and weeks, get your results from that GFP lab that we did a couple of weeks ago. Okay, the plates look really good, so I think you'll be pleased with those results tomorrow. Okay, so we'll finish up lab tomorrow as well. Okay, so I'll see you tomorrow for